Jennifer Listgarten from CS, and she's done work on bioinformatics, and look forward to hearing the talk.
Thanks. So I'm going to apologize on two fronts. One, I said to, when Bin first asked me, I said, I don't work in this area. And she said, this paper, talk about this paper. And so it's not quite fitting. But I'm hoping just that in any case, my goal here is to get you just interested in this problem and maybe how I think about it as a computer scientist, which is machine learning for protein engineering. And then the second thing I'm going to apologize for is the thing she asked me to talk about. I had a bit of an emergency this morning and I didn't get that much of it in. So, and I was in the middle of getting the deck ready and then something happened. So it's maybe a bit more scattered than usual, but please stop me if you have any questions or concerns. And so, okay, so let's get started. So basically, generally, I started this area around 4 years ago when I moved to UC Berkeley from Microsoft Research, where I spent all my years. And I'm sort of, my thinking is evolving on this. And some of the projects my group does are pure computational. And the point of this was to lead you up from our pie in the sky machine learning thinking to what we're actually doing with some wet lab collaborators on campus here and what we've managed to achieve, but I squeezed very little of that in. But so let me just motivate even the idea of re-engineering proteins. Probably when I tell most people now about proteins, can I close this? I feel like I can't see. People think of AlphaFold 2 and they immediately think that what I work on is predicting structure from sequence, which is completely not what I work on. And I will leave those massive engineering tasks to DeepMind 2, AlphaFold 2, to DeepMind. Of course, whatever progress they make feeds into the problems we think about. And I think they've made some really stunning progress. I'm not going to talk about it here, and I'm not even going to talk about structure prediction at all. I'm going to talk more about using machine learning data to train a supervised model and then use that model to design proteins in a kind of abstract way. And so, but there's two ways people think of or do protein engineering. One is to take existing proteins and then try to kind of change them, re-engineer them to achieve the purpose they want. So proteins are like kind of the workhorse of almost all of biology. So this one fluoresces naturally, but people engineer it to fluoresce more or at a different wavelength. And that might just sound like a fun thing to do, but actually this is like really important. It gets used all over academia and in industrial wet labs as well. Of course, everyone's heard of CRISPR-Cas9. This is a protein here that comes in the Pac-Man. It pulls the DNA apart, makes a cut, and then enables you to edit it. And people are also engineering that Cas9 to do all kinds of things like not to cut or to cut in different places and so forth. So the one I'm very excited now about for many reasons, including that I have a very close collaborator on campus, David Schaefer and his group, who's very interested in using viruses as mechanisms to deliver gene therapies, which I'm sure most people here have heard about. And so ostensibly, the focus of this talk is how to use machine learning to do this. And I promise I will say something about it, but I'm going to lead you through some of the things we've been thinking about before I get there. All right, and so I'm out of breath. I literally sprinted here. I didn't know it was a 25-minute walk from where I was. So I'm also a bit out of breath. But okay, so I'm not going to go through the very basics. I'm going to assume people know what amino acids are and that a protein is a string of can be encoded by a string of amino acids. There's an alphabet of size 20. You can see examples from that alphabet here that are drawn from our own alphabet of 26. And basically any string is syntactically a correct protein, but it might not fold, it might not be functional. And in fact, if you just randomly generate strings from this 20 amino acid alphabet, it's very unlikely they're going to even fold up into a structurally stable 3D thing, which is generally required for that protein to be of any utility. And so this is, of course, a vast combinatorial space. Proteins are often, in the hundreds of lengths of this alphabet. And so you can see just the sort of the sheer combinatorics get you near the number of atoms in the universe just for like the number of possible proteins for a fixed length that is within the normal size of proteins. So this is a huge, huge space. And so a lot of protein engineering is basically like, how do I navigate the space to get the thing I want to get when it's so massively large? And of course, my interest in this is to say, so this is the main way it's done right now in the wet lab without any computation. And my goal since I came to UC Berkeley was basically to think about what are ways that we can leverage machine learning to make this more effective. But who here's heard of directed evolution? Okay, it got the Nobel Prize a few years ago by these three folks. And it's sort of so simple and so easy to explain. I don't know how many Nobel Prizes that's true for. I wonder, actually. I think but so it's literally self-explanatory. Who has never heard of it, but thinks they can explain it from the name? Someone, but you're being shy. Plus, I don't have time to let you talk. Okay, so the way it works is you have to start with a single protein that already you know about and has like the seed of the thing you care about. So suppose you want this protein to fluoresce bright yellow. So you need it to fluoresce at least a little bit at some wavelength. And you can think of that as like a good parameter initialization for a machine learning model, because it turns out you can't just start anywhere. The initialization is super important here at the moment, probably forever also. And so suppose you start with that thing, and now you have to decide essentially in regular evolution, like the fitness is like our ability to reproduce and stay alive long enough to reproduce, and also be healthy enough to get our children to get to an age where they can healthily reproduce, right? Like that's natural evolution. In directed evolution, we're directing it because we're actually specifying the thing we want selection for. And so we actually going to create a population of things through some diversification mechanism in the lab. We'll start with that one protein. And then basically you can imagine just like at every position with some probability, you're going to mutate it at random, such that on the whole, there's some set of maybe 2-3 mutations from the original one you started with. And that's a diversification step, which gives you a variant pool. And then from there, you decide what you care about and you select out the ones that are not behaving very well according to that property. So if it's not fluorescing at all, you have to measure the thing you care about. And that's actually a big bottleneck here, because typically it's actually very hard to measure what you care about in biology, but fluorescence is quite easy. So you take, let's say, the top 10% according to the thing you care about, which you measure or at least a proxy of, and you throw out the rest. And you keep a pool of improved variants, and then you keep going until you hit some sort of convergence. And hopefully when you converge, you have what you want. And so this is very simple. And so, there's a whole, at the level I've described it, but there's a lot of complexity in how you measure what you want and also how to think about that diversification step. These are very important, hard problems, but abstracted for us kind of computational people, it's quite straightforward. So my goal was to say, can we work synergistically with this kind of idea and either get the same results with fewer iterations? And that can be super important because my collaborator, he works with like non-human primates in the lab, and like that's like has ethical concerns and costs and all kinds of stuff. So if you can just have the number of iterations from 6 to 3 on this like really, kind of crazy experiments, that's like really, really useful. And then alternatively, there's a number of things that just can't be done like this. People just don't have success and they get to a convergence of this algorithm and they don't have what they want. And so I'd like to both make it faster, and I'd like to be able to get us to places that we can't currently get to. And so when I first started working on this, I used to say, there's 2 places you might think to put in machine learning here on the face of it. And the very first obvious one is to replace the screening assay, this thing where you measure the fluorescence or the binding or whatever, with a machine learning predictive model. And so I definitely, if you want to do something smart in the space, you're going to have to do that. I used to say, I don't care about prediction. This is commodity by now. Go get your data, go get your scikit-learn and like train it. And if it works, it works. If it doesn't, it doesn't. Like I'm not going to have my lab focused on building predictive models, but I stand corrected in the sense that it turns out there's a bunch of interesting problems here about how to pick suitable inductive biases for these models for proteins, and also how to think about uncertainty and whether it's calibrated for these, especially as you move towards extrapolation space. So I won't talk very much about those depending, wow, how far we get. Do I get extra time because we started late? Okay, so what time do I have until? Okay, so the one I got more interested in initially, but these of course are interrelated, is like, if you think about what directed evolution is doing, it's doing like a random greedy walk, right? It's like, let's randomize to a pool of variants and then move to the best one and then do again. And so you can, kind of think rigorously about what kinds of functions you could have success on if you move in that kind of greedy stochastic way. But it stands to reason that if we could understand something more about particular landscapes that we're moving through, then maybe we could do smarter moves than these kinds of greedy stochastic moves. And so that's how I kind of first got interested, but now I'm interested in all of this and much more. So let me tell you a little bit about the story as it started for my group when I moved here with my first student, David Brooks. So we started saying, suppose we had a little bit of supervised data and we could build a model. And this is actually what happens for the actual instantiation of this with gene therapy that I'll tell you about. They gave us some data and they said, this is what we care about. And in general, you care about many things, actually, especially when you get into therapeutics. You don't typically care just does it hit the target I want. You care about toxicity, clearance from the body, and so forth. But I'm just generally, so far, we're going to usually just think about one property. Now, suppose you have this data and you train some model. I've put a neural network here, but it could be whatever. However, in practice these days, neural networks actually work really well on a lot of problems. And now the idea is that instead of training the model as usual, I want to assume I've trained the model, and I want to specify the output that the desired output, like maybe binds this target high enough. And using the already trained network, I want to, in a sense, learn the inputs that will give me what I want. And so on the face of it, this is just a pure optimization problem where I'm optimizing the function defined by the neural network. But actually, this is actually, and people have thought about it this way and published papers, and they're more or less complete nonsense. So why are they complete nonsense? Any ideas? Am I allowed to drink? Is this like legal? Can I do that? So, right, this explains it. What's the problem? So there's an analogy in computer vision to the problem I've described. The problem I've described is how do I get a protein that has the property I want? And the analogy is many years ago in computer vision, people would train these massive state-of-the-art neural networks, and they'd say, I want to know what my neural network thinks a banana is when it, even though it also seems to know what a zebra and an apple and a pear, blah, 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 blah. So what they do is they have a multi-class thing for each class there in their thing. They set all the outputs to 0 except the banana node to 1. So it's different because I care about regression. They're doing classification, but it's basically the same. And then they do back propagation to do gradient descent to minimize an objective that gives them the best, highest banana output. And they ask what that image looks like. So what does that image look like? It looks like abstract art. So you say, it's not crazy, right? Like I can see how this might make sense. It has some curves, it has some yellow. But if I were to send this to the 3D printer, this is not gonna, like, no one's gonna think this is a banana, right? So what has gone wrong essentially is that we have these pathological blind spots in these highly over-parameterized models, probably even in very simple models actually, but especially in these over-parameterized models. And so what's interesting is this is not an adversarial, like it's not a game, it's not adversarial when I design a protein, right? I'm just saying I want my protein to have this property, but it actually kind of behaves in an adversarial manner because you're pushing it to the corners of the space to maximize this thing. And so sort of in effect, it behaves almost as though it's like adversarial, getting to weird corners that just don't make sense. And this is actually in some, like all real problems, this is an impossible problem to deal with, but I think one of our contributions is to, one, recognize it and think about it and at least propose some formalisms that would allow us to reasonably mitigate it. But I think progress can always be made. And at the end of the day, you want a formalism that enables you to think cleanly about this and bring to bear your domain knowledge. So that's sort of how I got into this space of machine learning and protein engineering. And so I'm going to, I totally switched the talk around and then I ran here. So let's see if I can understand my own logic of these slides. But let me just tell you, if you think about the problem that way, like let me reiterate and explain a bit more what some of the problems that emerge here. And this is part of the reason I like working at this intersection because, there's all the standard problems in NLP and vision, but I feel like when you go into these scientific domains, you get to think about things that sometimes also have application and analogy there, but you get to think about slightly different problems. And so kind of the key issue here, right, is this natural tension between how do I take a trained model and kind of, let's suppose I want to maximize a property, and I'm basically asking it to extrapolate. And I think we all know that we can't extrapolate like arbitrarily, and that's essentially these pathological corners that emerge. And so the question is, how do I navigate this sort of ability to extrapolate somewhere useful by understanding where I trust this model, and how can I think about where I trust this model? And so we often see these sort of Gaussian process regression curves like this, where the, I only learned these terms this year, epistemic uncertainty. I had to write them on my whiteboard because I couldn't remember what they were. But the epistemic uncertainty is the uncertainty you get just by being far away from the data, right? Usually we think of like the sensor noise, instrument noise, which is the aleatoric. Correct me if I got them wrong. I think this is right. And so in some sense, we really care about estimating the epistemic uncertainty in some reasonable manner and leveraging it throughout this process. And so I'm just listing here, these are basically things we've started to do in my group that touch on all of these through various of my students. And so this is, of course, related to causality. If I had the true causal model, this wouldn't be an issue, but I never have the true causal model. And we've not started to think about this in terms of methodology from causality, but there, perhaps there's opportunities there. And okay, right. So this is also related to this epistemic uncertainty that I mentioned, and like how can we estimate it well and use it to do these kinds of things. And then, as I mentioned, I have one slide versions of all of these at the end if I have time, which I probably won't. But also this, as I mentioned, if I am going to invert this model, but I'm also the one that gets to build it, then I might want to think, and we started to think a lot about this, what are good ways to think of inductive biases for these models? And just as an example, if you think about dropout or convolution, or like, these are all inductive biases, right? But in proteins are these discrete inputs, and we know actually that it's sparse in the features, which are just the single amino acids and pairs of amino acids and triplets of amino acids, sort of first and second and third order. And if you try to regularize at the level of the parameters, you can never capture that. And so there's this sort of, it's hard to think about what's the right way to induce these inductive biases when I parameterize it with this giant neural network that doesn't map to that. And then, so this is the one I'm going to try to talk about as it pertains to this collaboration we've been doing for a few years, which is that the way I've described it so far is to say, I'm going to tell you what outputs I want, and you go find me a sequence, right? And so, of course, everything's imperfect. And in practice, what we want is to hedge our bets and have a set of sequences there. And so there's actually one of my students is very interested in actually trying to characterize how we should think about hedging our bets in this sequence, which is very related to batch active learning. But it turns out that in protein engineering, we cannot often go back to the collaborator and say, please, print this particular sequence. Because what they actually do is, instead of just creating individual sequences, like, you could imagine I have an Excel sheet and I say, please make this sequence and this sequence and this sequence. Like they don't typically, well, they sometimes operate that way, and it's almost certainly the way of the future, but there's a lot of very interesting things being done now where it's more constrained than that. And so what you actually do is you design a distribution over the sequences, and the thing you get to give back to your wet lab collaborators are the parameters of that distribution. And I'll say more what I mean about that in a second. But if you hear the word library design, like a library in biology is basically a distribution over sequences defined by some knobs that you can tell your collaborator to set in this particular way. Right, and so this is the culmination of several years of work with David Schaefer's lab and Bonnie, the postdoc, and David, my first student, where we're using some of these ideas to actually advance some of the important parts of AAV engineering for gene therapy, and I'll explain more. Okay, so I think I already said this, but right, so library design is a distribution design, and that's to be contrasted with designing individual sequences, which is the way you would think about this, or an individual image. Instead, it's like a distribution over images, and that's when we cannot feasibly specify each individual protein in the library. And so let me instantiate that with actually the real example that we use, which you'll see is super, super simple. So this is what we actually get to do in the real life example that I'm going to tell you about. And I mean, this is so real. This is actually like stuff that's through FDA approval for blindness therapies. This isn't like real. We did something in the wet lab here on campus. This is really impactful stuff. So in this case, it's going to be a sequence of length 7 from the amino acid alphabet. And the only thing we can tell our collaborators is the marginal distribution of each amino acid at each position. And so that's actually a pretty rudimentary control knob, and yet is actually good enough to do some really important things. And that's just the one we're focused on. There's quite a large set of these that you could think about as well. And sometimes there's a trade-off between cost and scale. So to the extent we want more precise control, usually we cannot generate as much or sometimes measure as much. And so even if the technology is there to do something more specific, we don't always do that. It's true here in particular. So if I'm interested in library design and I'm thinking about protein engineering in the context of this sort of invert my model, while taking into account the pathological areas of the space, it's useful to think about that optimization problem in a different way than you might usually. So how do we usually write an optimization problem? You might normally think this is the neural network point prediction or something based on its cumulative density function to account for uncertainty. And then you might say, I want to maximize this thing. So I'm now going to search through protein space where X is a protein. And so what we did actually before we were doing library design for other reasons, but it turns out this is the perfect thing to do for library design, is we said, instead of searching over individual sequences, let's search over a distribution of sequences. And I think of this as now like a spotlight in protein space. So it's a bit of an abuse. I tend to think of it as like a Gaussian. It's an abuse because of course this is discrete, but I think it's a helpful visual to think I'm going to do some optimization that starts off with some Gaussian, and I'm going to move it around instead of following a trajectory of individual proteins. And when I convert the problem to this, it becomes an expectation with respect to that distribution where I'm trying to find the theta, which is sort of like the mean and the covariance. And I'm going to move it around and change the mean and covariance to get to a part of protein space that has the property I want, and then I can sample from it. So can you just do this? So I'm not going to go through any proofs, but you can probably see somewhat intuitively that when this distribution, parameterized distribution, if it's high enough capacity to put all of its mass on the single protein that maximizes things, then they're equivalent. Doesn't mean you can find it. It just means the formulation is equivalent. And so you can always do this. And you might actually, one of the nice things, and I'll get to this in a second. Well, actually, I won't say it yet because it's on the next slide. But does this make sense? So we're converting the problem from just searching for an X to searching for a distribution over X. And now, originally, we weren't doing library design, and we were actually just kind of using this for reasons I'll mention in a second. But if you have a library that has particular parameters, you can just actually model exactly those parameters here. So it's also convenient, right? I can just say, only allow this distribution to have the parameters for the marginal distribution at each position. And then I naturally get out the parameters.
Is P a known parameter distribution, probability distribution? So how does it look like? Is it based on the library that you're looking at, or?
So this is just, I mean, so if sequences were real valued, I could think of it as a Gaussian. Since sequences of amino acids are not, it could be a hidden Markov model. It could be a variational autoencoder. It could be a mixture of hidden Markov models. It could be multinomial, things like this.
So you have a description of the probability distribution P. Is that?
So we're going to find the parameters that we're going to try to find the parameters that maximize this expression.
Okay.
Yeah. So that's like, you start with some distribution over proteins, and as I'm not going to go into too many details about the algorithm, but it moves around and then it converges. So this is nice because it's sort of directly relevant to library design, because this is essentially how we think about libraries in biology, if we can map the experimental design knobs to some parameters that we can actually fit. It's also, there's some other reasons that it's nice. The one maybe that I'll highlight that I think is just kind of cool, and the reason we first started to do this, is that I've actually brought in the language of probability into optimization. and like in a kind of weird way. And so you're like, okay, well, why is that real? Like, does that bias something? And so I won't go into too many details, but it turns out once we're in the language of probability, it enables us to formally and coherently bring in domain knowledge to help us rein in from going into those pathological regions. And that's actually why we first started to use this. And in a sense, you can define like probabilistic trust regions based on, for like an example would be, there's huge databases of all known proteins that stably fold. So if you could build some sort of, and people can build a density model over those, now you have a density over all known proteins that fold, which is a pretty good way to rein in your exploration to prevent it from going crazy places. So we have a paper out where we explore it's a ICML paper where we kind of get into this, and in particular how to think about these soft trust regions to mitigate the abstract banana problem. And I won't go into it. It's an iterative algorithm, has some few challenges, and it has some nice intuitive correspondence to directed evolution itself, which I won't go into. And it has some connections to some other technical areas. So let me just get, because I'm running out of time, to what Ben said. Maybe it'd be nice to talk about this. So basically we've been thinking a lot about all of these ideas and kind of picking one off at a time, and sort of pie in the sky methodological papers that I think of as almost just thought papers, but they have empirics to back them. And I don't trust anything until I'm actually doing something real. And so this has been a multi-year project with many rounds of experiments, a ton of discussion to understand each other. And we finally got the preprint up just about a month or two ago, which you can check out. And so this is all centered on gene therapy using the adeno-associated virus, AAV. There's a picture here. It's actually a single protein that gets repeated in a sort of symmetric space. And the thing we get to engineer, you can't really see because the resolution's bad here, but the little red bit is a 7 amino acid length insertion into the capsid, which is like the outside of the protein. And they identified this position as a place where if you just change the 7 amino acids there, you get quite wildly different results for things you care about. So in some sense, it's a good place to induce variation to try to achieve all kinds of engineering goals. Right, and so what is gene therapy? It's basically you want to use this virus, you want to put in imagine you're missing some enzyme that you need to like live well. And so you want to deliver that enzyme to some particular tissue that's missing it. Then you need to basically create it, put it in the virus, and then you need the virus to infect exactly the right cells and not the wrong cells. And that's kind of one of the main ways that this gets used for gene therapy. And so there's actually quite a number of interesting engineering opportunities here. And we tackled the very first one, which is at the sort of root of all that follows when you use it in this manner. And it has to do with packaging. And so the way people do these libraries, I think it's on the next slide. So this is this thing I already explained to you, is we're going to get to design the probability distribution at each position over the 20 amino acids. And then they're going to put that in, and it's going to appear in all these repeating units. And depending what's there, it will actually not even be viable. It won't even fold up into a reasonable capsid. It's just complete garbage. Or it will fold up and have different properties. It will infect this cell in the eye, or it won't. It'll go through the blood-brain barrier, or it won't, and things like this. And right, and so Dave has a company that's just gone through FDA approval, as I said, and it's actually to help with like a blindness disease in the back of the eye, and they had to engineer it to get to the back of the eye. And so the problem is the state-of-the-art library they use for this. So what they do is they create like this library that has this huge diversity, something like 10 to the 6 variants, and then they screen it to get to the eye. But actually half that library is completely wasted because it doesn't even fold up and function. And so in some sense, they've like lost half their ability to find good things, and they've wasted this sort of precious ability to measure things. And that's just because they don't know how to design a library such that it has like, what do you want? You want great diversity in order to be able to have as much sort of raw material that has some chance of getting to the cell you want, but you also want it to be functional in some very general level that's a precursor for all downstream criteria. And so the challenge we had was how can we get a library that packages, in a sense, twice as well as the state of the art while maintaining the same diversity. So I think I'm out of time, so I'm just gonna try to very quickly tell you how we did this. So the first thing we do is they generate with the standard state-of-the-art library that actually wastes half of itself. We build a predictive model and we check in the lab that this predictive model actually works. It actually does what we think. And then we make use of kind of this optimization I told you about, but we add an entropy term to it to control the entropy. And then we can trace out a Pareto optimal frontier that's trading off this the diversity of the library with the overall expectation that it can actually package and function, sort of generally be fit. And so the library that's state of the art is down here, is like complete, which is like just crap. It's like half of it is wasted. And then now we can start to rationally think about how to pick a good library. And so that's essentially what we've done. We picked a library, and I don't think I have the numbers here, but we can show that in a downstream selection that had nothing to do with anything here, because it was for a specific cell type, we can infect that cell type in the brain about 10 times better than they could with the regular current state-of-the-art library. And so the idea is to get a generally good library for different downstream tasks. And we showed that for one of those downstream tasks that we've not optimized it for, we do much, much better. Okay, so sorry, I threw a lot at you. I definitely don't have time to go through anything else, but if you're interested, this is some recent stuff from my group. And I'll just say, yeah, I've been here at Cal for 4 years, and I'm so glad to have all my amazing grad students. Thank you.
Thank you, Jennifer. So we need 2 more chairs and we'll have a panel and we can get all the questions going. The other